We start now, right?
Yeah.
Yeah.
Yeah, it started.
Mbak Sylvia, ini saya ngomong duluan ya?
Ya, boleh dibuka.
Oh,
Nanti saya untuk introduction dari perusahaan aja.
okay.
Yeah.
Thank you, ladies and gentlemen, for coming to the webinar series today. This webinar is held by Science Werke together with MGI. So the webinar entitled as a fight against COVID-19, massively parallel sequencing for SARS-CoV-2 detection and surveillance. So in this webinar, it consists of three speakers. So, four speakers. So the first speaker is Ms. Sylvia Sanse. She's our product manager for genomics, and she will explain you about the MGI, and probably because most of the audience is Indonesian, so we would like to Sylvia would like to explain in Bahasa Indonesia, so therefore the audience can better understand about the content of the presentations. And afterwards, there will be presentations from Dr. Zhizuan Li, the MGI product manager, about the NP-seq technology. So he will explain about the technology behind the MGI, massively parallel sequencing, NGS sequencing, and about the platform behind it. After that, Dr. Yao Jiang, the MGI senior product manager, will explain you about the MPS of SARS-CoV-2 sequencing. So it will be very interesting, yeah, because now it's the genome sequencing for COVID-19 virus. It's becoming popular to see, to track the epidemiological and everything, yeah, related for it. After that, Dr. Wei Tian, our MGI FPS, will give a report the interpretations of the sequencing. And then in the final, as usual, we will have a question and answer that will be held by me and Ms. Tika for you to have questions and also comment about the presentation that has been given by all of the speakers. So without wasting time, I would like to let Ms. Sylvia Sansa, our product manager, to give the presentation. So this time is for you, Mbak Sylvia.
Oke, baik. Terima kasih Pak Sony atas pembukaannya. Selamat pagi Bapak-Bapak dan Ibu peneliti sekalian. Terima kasih atas kehadirannya pada webinar pagi hari ini. Webinar pada pagi hari ini merupakan kerjasama antara MGI dengan ScienceWorks, dan saya akan mencoba memperkenalkan ScienceWorks dan MGI secara ringkas dan kemudian terkait dengan tujuan dari diadakannya webinar ini. Jadi ScienceWorks merupakan sole agent untuk MGI di Indonesia, di mana untuk ScienceWorks sendiri kita memiliki beberapa head office affiliasi di Myanmar, Singapura, Indonesia, dan Malaysia. Dan untuk di Indonesia sendiri, kantor pusatnya berada di Palmerah Barat 25 di Jakarta Barat. Untuk beberapa cabang kita punya di Surabaya, kemudian di Bandung, kemudian di Jogja, dan beberapa representatif area. Antara lain di Makassar, kemudian ada juga di Malang, dan lain sebagainya. Nah, untuk ScienceWorks sendiri, kita sebenarnya ada 6 divisi. Jadi MGI ini merupakan bagian dari divisi genomik, sedangkan dari ScienceWorks itu sendiri kita ada divisi proteomik, kemudian ada analitik, kemudian ada applied science, cell biology, bioservice, dan juga diagnostik. Jadi kami memenuhi kebutuhan untuk peneliti dalam aplikasi apapun. Kemudian kami juga dilengkapi dengan tim FSE dan juga tim FAS yang membantu after sales support kami. Kemudian untuk BGI sendiri, jadi BGI ini merupakan perusahaan besar yang sudah lama bergerak dalam bidang genomik. Jadi basicnya adalah di pelayanan untuk sequencing. Nah, untuk BGI sendiri ini sebenarnya sudah cukup lama berkecimpung, jadi lebih dari 20 tahun berkontribusi dalam human genome, terutama human genome. Nah, untuk MGI ini merupakan anak perusahaan dari BGI. Jadi MGI ini pusatnya adalah di Shenzhen, Cina. Jadi di MGI ini salah satu kegiatan utamanya adalah untuk develop dan produksi alat, terutama untuk alat-alat sequencing. Nah, pada hari ini kita akan sama-sama mendengar paparan dari MGI terkait dengan inovasi teknologi sequencing yang mereka miliki, terutama untuk aplikasi COVID-19. Nah, untuk rangkaian webinar kali ini, sebenarnya untuk webinar ini kita tidak hanya pada hari ini, tetapi nanti akan ada beberapa webinar lanjutan, di mana di setiap webinar kita akan memiliki topik masing-masing. Nah, yang kami highlight di sini adalah dari total solution dari MGI, di mana kita memprovide mulai dari pengerjaan, kemudian sequencing, hingga kita mendapatkan report yang bisa direpresentasikan atau bisa kita ambil manfaatnya. Ada beberapa aplikasi yang akan kita provide untuk webinar selanjutnya. Nah, ini adalah salah satu contoh report yang bisa di-provide oleh MGI ini untuk pathogen fast identification. Jadi kita bisa melihat identifikasi bakteri, kemudian kelimpahannya. Kemudian untuk bakteri juga sama, jadi kita bisa melihat serotype identification, kemudian gen resistennya, virulensi. Nah, ini yang akan kita bahas di next webinar. Nah, ini adalah plan keseluruhan dari webinar. Jadi untuk bulan Juni ini topik kita adalah Atoplex for COVID. Nah, di next bulan depan kita ada lanjutan dari webinar kali ini, ada pathogen fast identification, di mana kita akan membahas tentang microbial identification dan resisten, kemudian bagaimana workflow-nya. Kemudian di bulan September kita ada reproductive health, di mana kita akan mempelajari tentang NIPT, kemudian PGS, dan bagaimana interpretasinya menggunakan sequencing. Dan yang terakhir, di bulan Oktober kita punya fokus di cancer research dan service analysis. Nah, kita fokus pada hotspot gene detection, kemudian BRCA1, 2 detection, dan juga untuk lung cancer. Nah, itu mungkin summary dari kami sebagai marketing dari Soundsworth. Saya kembalikan ke Pak Sony untuk melanjutkan webinar kita. Terima kasih.
Cynthia for the informations about the SARS-CoV-2 and about the genetic activities related to MGI activities in Indonesia. So for the question and answer, if you have question, you can type in our chat sessions, and please give your name and the speakers who you want to be asked. Yeah, please type your question concisely, not very long. You can do it in Indonesian. We will translate it for you. It's no problem. And also please do it in a short manner, yeah, in a concise manner. So for the next session, I will introduce you about the second presentation about DNB-seq technology and platform introduction from Dr. Ziswan Lee, the MGI product manager. The time and the opportunity I give to you, Mr. Ziswan Lee. Hello.
Hello, everyone. This is Li Zixuan from MGI Product Center. And today I'm going to talk about DNB-seq sequencing technology. Okay, sorry. Slides doesn't work. Okay. First, I will make a brief introduction of this technology. It's including DNB, what we call DNA nanoball. It's a highly amplified single-strand DNA, and also a technology for the full cell called pattern array. Okay, first we got a single-strand circularized DNA, and we used RCA rolling circle amplification to get a structure like this. We call it DNB, DNA nanoballs. Then we will load it on a flow cell. And this is what a flow cell looks like. And all this technology will make the data output more accurate and also can decrease the duplicates and avoid the index hopping. And later I will explain the detail step by step. Okay, the first type is amplification. We use RCA, rolling circle amplification. We can imagine a single strand DNA as a single match. So when we lighten the match, the single match, it will make a very weak light. And if we want to have a better signal, we need to copy the match to amplify the DNA. Then we have 300 to 500 copies, a piles of match. When we lighten it, it will get very strong signal and will be easily captured by our camera and transferred to our Atgc sequence. And why we use RCA, not the traditional PCR? Because the PCR has some limitation. For example, it can accumulate the error. Like if we have an error here, a mismatch here, and this error will pass to the following cycle. It will be the template of the following amplification, the base pair. So all these branch will have the same mistake. But if we use RCA, all these circles, the amplification will use the original single strand DNA as the template. So if we have a mistake, and this mistake won't accumulate to the next cycle. So it will have a better accurate and also can avoid the index hopping. And after we got the DNB, we will load it on the flow cell. And this is what the flow cell looks like. It has millions of spots regularly located on the flow cells. And the DNB will attach on each spot with electrostatic interaction. And we will also add some protein to embed the DNB. After we fix the DNB, we'll add some DNGP with a fluorescent dye and use laser to make it lightening. And we will use camera to capture the picture like this. This is the raw image. And we will distinguish each spot and translate it to Atgc. Then we will correct the signal. There will be some and use the signal, and we will finally decide which base it is. And this will be the final report. Each spot will have a sequence, and the millions of spots will get the whole file, called FASTQ file. And we also make some optimization on the algorithm, the base calling algorithm. The first one is optimized image processing. Now we have a more accurate signal positioning and identification. And second one is a very important function called copy number correction function. And that just means we can use the first 10 cycles as the reference to correct the following cycles. We know the fragment has a different size. For the shorter one, if we have the same time for amplification, the shorter one will have more cycles of amplifications. Then it will have a stronger signals. So we use the first 10 cycles as the reference to balance the signals. Then we can normalize the signal between DNBs for better signal separations. The last one is to optimize Atgc clustering. That is when we receive the signals and to distinguish which base it is. It can increase the capability of processing more complex libraries. For example, unbalanced libraries like ATOPLEX. Okay, after this optimization, can have fewer errors and also a high mapping rate. Okay, now I will make a brief introduction of MGI sequencer portfolio. We have different ranges. We have all range of throughput of sequencers. For low throughput, we have DMB65. It's a very small sequencers. It can support targeting sequencing, and it has a very fast performance. And we also has nearly 20,000 gigabyte throughput DMB6 T7 and TX, and that is for population genomics research. And today we'll focus on DMB6G50, which is the recommended platform for pathogen detection. Okay, here are some major features for DMB6G50. It's compact, flexible, fast, and versatile. It is designed as a benchtop sequencer, only takes less than 0.5 square meter. It has a large range of data output from 10 to 150 gigabyte. And we have two choices of flow cells, the larger one with 5 million reads and the smaller one with 100 million reads. And also 4 different read lengths. We can combine the read length with the flow cell, 2 different flow cells. Then we will have multiple choices for different use, like cancer early screening or pathogen detection, also the whole exome sequencing, whole genome sequencing. And another very important point is it's very fast. We have improved this performance. Now we can finish P100 with FCS in 20 hours. And that is the recommended read length, the sequencing mode for 8-plex. And also the SE50 for metagenomic sequencing. If you have a very emergency demand, you can finish NGS sequencing in 6.5 hours. Here are some details about 2 different flow cells. The first one, the larger one, has 500 million reads for read length, and the maximum data output is 150 gigabyte. It has a high data output, which means it can support mixed library. For example, if you have a capture-based panel and also the multiplex PCR-based panel, you can mix 2 different library and test it in one run. And also because of the high data output, we'll have a low cost per sample. Here are 2 solutions for pathogen detection. One is metagenomics. For example, it will take 20 million reads per sample. And if we use FCL, we can test 25 samples one time. Another one is 8-plex PCR panel for COVID-19. It depends on the CT value, depends on the condition of the sample. If the sample is in a good condition, we can test over 60 samples. And for FCS, it has only 100 million reads, 3 read lengths. It has lower data output, but it has very fast sequencing performance. Also, because of this has a low throughput, it has a low cost per run. It also supports both 2 applications for pathogen detections, but it has very fast sequencing speed. Here is the flow cell specification. I already highlight the recommended sequencing mode for 8-plex sequencing. You can see it can not only just for a fast runtime, it also has a very high quality. Above 85% data complied with Q30. Here are the performance. We test the human cell line 12878 sample with 2 different library preparation methods. One is PCR based and one is PCR free based. And we also compared it with our another platform, DMB6G400. We can see about 94% data comply with Q30. Is very high quality of data output. And also there are some parameter about mapping rate, unique rate. We have a standard. You can see both G400 and G50, the performance are far above the standard. And also the mutation detection. Some customer doesn't care about the Q30 or the mapping rate. They only care the final report, the detection result. You can see the both precision and sensitivity. G40 has a very great performance. And we also have a total solution cover the entire sequencing workflow with not just provide the sequencer, we also provide the, for example, DNA extraction kit and library preparation kit, and also the automated sample preparation system, also the bioinformatic analyzer. For different uses, we have the related reagent kit. Here I want to share some cases. The first one is from Thai CDC. We know for CDC, they will face some problem, like they will receive some sample in a bad condition. For example, the sample's CT value is nearly 40, close to the threshold of RT-PCR detection. They cannot use RT-PCR to detect this sample. And also there another condition, another situation is the RT-PCR results are not consistent. For example, first time negative and next time positive, they cannot confirm the result. Then they will use high throughput sequencing to confirm the final result. And they use G50, and they prove that our platform can detect this trace viral load viral sample. Here is another case from Korea. It's made by Korea Center of the Korea CDC and Seoul National University. It's a research about transcriptome of COVID-19. They used both our platform and the third generation sequencing. And finally, they found that the depth of DNB-seq allowed us to confirm and examine the junction on an unprecedented scale of COVID-19 genome. It proved that we can have a deeper research on transcriptome. Here is cases from China, and this happened on the end of last year. There is a female passenger from the UK and tested positive for COVID-19. And Shanghai CDC used our platform, DNB-seq G50, also our 8-plex PCR panel, and they recommended a sequencing mode FCLP100 to test this sample. And finally, they got this strand from the new variant from the UK, and this case was confirmed as the first imported variant in China. Okay, that's all. That's all I want to share. And this platform compact, flexible, fast, and versatile. This is all about DNB-seq G50. And thank you for listening. And next, we'll let Dr. Jiang for the next sharing.
Hello, everyone. I'm Jiang Yao, the product manager of pathogen detection products. Today, I will introduce the large-scale automated PCR-based genome sequencing solution for COVID-19 to you. I will share the following information. We participated in the COVID-19 pandemic prevention in many European countries, such as Sweden, France, and Denmark. In Karolinska Institute, Sweden, we set up a high-throughput sequencing system of 10,000 samples per day for virus variation and traceability. Our Huoyan laboratories are distributed in Italy, Spain, Serbia, and so on. They are used in rapid COVID-19 detection with a maximal throughput of 10,000 samples per day. In Latvia, MGI Technology as Latvian scientists to discover more than 20 unique mutations of COVID-19. MGI instruments are also widely used in pandemic prevention in Kazakhstan, Angola, Ethiopia, and other countries. Here are some COVID-19 related literatures based on MGI platform, which were published on some top-notch magazines, such as Nature, Cell, Lancet, and so on. MGI platform has been widely acknowledged. Next, let's introduce the COVID-19 sequencing and tracing with Autoplex platform. There are 4 features of the Autoplex platform: large scale, less label, highly automated features, wide use. Compared with RT-PCR, the advantages of sequencing is being able to get full-length genome and monitor mutation. For SARS-CoV-2 detection, we are recommending 2 sequencing strategies. One is metagenomic sequencing, which is to sequence all nucleic acids in a sample and possible co-infection and secondary infection. Since the pathogen load is generally less than 5% in human samples, for no RNA input, more than 100 million rates is suggested to achieve high detection sensitivity. The other is multiplex PCR sequencing, which is more focused on SARS-CoV-2. A SARS-CoV-2 primer pool is designed to capture the virus at library preparation. So much less rates are required for sequencing while increasing the detection sensitivity. In addition, this strategy could get full length of a virus genome. We have total solution from sample extraction to library preparation, sequencing, and data analysis. At first, we recommend the MGI secondary generation nucleic acid extraction kit to extract RNA. Then we use our Autoplex PCR panel to construct the library. We choose different sequencing platforms for high throughput sequencing, including DNB-seq T7, DNB-seq G400, and DNB-seq G50. Per under 100 rate lengths is recommended. We can use in-house pipeline or Megaboard to analyze the sequencing data. Generally speaking, the entire time is about 2 to 4 days, and we can test 400 to 20,000 samples per week. 2 to 15 people are needed, mainly based on the number of samples. We know that the SARS-CoV-2 virus is a single-strand RNA virus, and the length of the virus is 29.9 kilobase pair. Our multiplex PCR kit can cover the full length of the virus genome. Therefore, it can assemble, analyze, and trace the virus genome. We need to perform reverse transcription and 2 rounds of PCR experiments to obtain an amplicon library, and finally perform high throughput sequencing and genome assembly. The multiplex PCR kit covers the full length of the virus genome with a total of 273 amplicons, and the average length of each amplicon is from 100 to 200 base pair. This kit could get full length genome as low as 10 copies input. The kit could quantify copy number of SARS-CoV-2. The most important thing is this kit could detect India, UK, Brazil, and Africa SARS-CoV-2 strains. In order to compare the sensitivity of qPCR and the multiplex PCR methods, we use the 7 gradients of a reference sample to perform serial dilutions. From this table, we can see that when the gradient is diluted to D8, the red number. Yes. qPCR has not detected the virus, but the 100-time coverage of SARS-CoV-2 genome is more than 4.8%. Therefore, multiplex PCR technology has high detection sensitivity and can obtain the full length of the viral genome. In Karolinska Institute, Sweden, the testing process, including sampling, delivery, MPC, RNA extraction, library preparation, qPCR for diagnosis, and the sequencing for phylogenetics analysis. Last year, they have conducted diagnosis for more than 30,000 samples 1 week and assembled more than 20,000 COVID-19 sequences. This work started from May. Karolinska Institute construct a large-scale population SARS-CoV-2 tracking system with autoplex solution. They have 8 MGI SP 960, MGI SP 100, DMB-Sec T7, and 20 qPCR instruments. 3 people could test 3,000 samples per week. If 2 more people, the throughput could increase to 5,000. They have applied the multiplex PCR-based sequencing method in Swedish SARS-CoV-2 genome study, compared with the conventional shotgun sequencing. This method is faster, more sensitive, and more cost-effective in sequencing SARS-CoV-2 genomes. Instead of randomly fragment the viral genome, multiplex PCR-based sequencing applied 273 pairs of primers to target and amplify the entire genome of SARS-CoV-2 by regions. After library preparation and sequencing, they assemble and amplify genome regions back to the genome and conduct phylogenetic analysis and SNV analysis. These null rates are spent to host or other pathogens. Based on our experience, only 3 million rates were required for a sample using multiplex PCR sequence. Among 20 and 10 sequenced samples, there are more than 1,600 high-quality assembled genome, which have more than 95% coverage with 100 times sequencing depth. We used the high quality COVID-19 sequences together with 1,000 randomly selected COVID-19 sequence from the globe to make such tree. We can see that the sequence assembled by us are mostly from the clade 20B. Smaller portions are from clade 20C. The University of Queensland used the autoplex tracing the SARS-CoV-2 by wastewater. 2 wastewater samples were shown to be closely related to multiple viral strains that originate from North America and Asia. This finding suggests the feasibility of this approach for phylogenetic studies on wastewater samples. The left case reports the entire genome of the virus are successfully assembled, and it was confirmed as the first imported British variant virus strain in China through traceability research. The right case report through following up sequencing of some twice infected patients. It demonstrates the existence of a reinfection and revealed the immune mechanism that may affect the reinfection. Each case comes from Latvia, Europe, with MGI sequencers. Their technicians successfully detect 4 British variants and trace to the source. Here are 2 academic papers about COVID-19 sequencing and analysis with Autoplex platform. We could provide different throughput programs. 2 to 4 people can finish 500 to 1,000 samples per week. 3 to 5 people can finish 2,400 to 4,800 samples per week. And 10 to 20 people can finish 10,000 to 20,000 samples per week. There are the ordering information, including the instruments, reagents, software, and so on. Summary. Autoplex solution enabled scientists to obtain the full-length genome sequence of SARS-CoV-2 at low cost and high capacity. Autoplex solution can process samples with a relatively low amount of virus genome. Autoplex can help scientists and institutions to carry out large-scale SARS-CoV-2 genome research. MGI can provide customized solution based on research scale and applications. That's all. Thanks for your listening.
Thank you, Dr. Yao Jiang, for your presentations. And the next will be report interpretation from Dr. Wei Tian, the MGI FPS. For your opportunity and time, I give you Dr. Yao Jiang.
Hello? Yeah. Can you hear me clearly?
Yes, we can hear you.
Yeah, cool. Yeah, my name is Wei Tian. You can call me Taylor for short. And I'm very glad to have this opportunity to talk a little bit about the report of the IGVD. From the start, we will have a brief picture of this platform. Actually, it's quite a straightforward platform which can give you very convenient analysis from the input of your, let's say, FASTQ file to the final report. Let's say some like basic analysis result, or the mutations information, and the, let's say, phylogenetic analysis results. And the usage is quite straightforward. All you can do is to make a list of your input FASTQ file and press a couple of buttons and you will find the result. Here is the system module. I will give you more detailed introduction about this part later. And I just want to give a brief introduction of some key features of this system, like the sample management, task management, system management, and the result viewing. We can start from the pipeline of this analysis. The analysis input, the input data is the raw reads. And after the quality control and sequence alignment, we got the BAM file, which is very useful for like many following analysis, like advanced analysis and other sort of other analysis. From the BAM in the Atoplex, we can have some basic information like the coverage calculation and the various concentration, quantification, and the mutations. And also based on the mutations, we have some assembly and some like downstream analysis based on the assembly, like the clade analysis and lineage analysis. So the first step of this whole process is to input your samples. And you can have, we provide you a CSV template. You just input your information based on this template, and you use the input introduce your data to the system, and then press the analysis, and all the analysis download the downstream analysis will be triggered. Yeah, and after the analysis, you will have some information like you can have a look about what we got from the analysis. Like the upper part, you can click the tasks, which is highlighted right. You can click the tasks, and then the result will be displayed in your screen, and you can have a brief idea about this analysis, like the one I highlighted, like the positive one. We have some criteria for the positive, which is based on like more than 100 cases in our previous projects and analysis. And following by that, you can have the log and report and PDF. Like the PDF is all the reports will be that can be downloaded as a PDF format. And then after that, you will have like some advanced analysis based on the like, let's say, basic analysis. You can have some like summaries, variants, and you can also download your genomes, like assembly genomes based on the variants. For the summary, we can have like some very pretty standard output. Like I just want to skip this part to save the time. And for the variants, also the result is very standard. I just want to highlight the analyze genomes. Like after you download the genomes, you can upload the genomes to the next strain or other like COVID related databases or analysis systems. It will give you some more ideas about this genome, like the variants, like the tree information, like what clade is belongs to this kind of information. But IGV, is quite safety, let's say safety, because if you want to keep your data, if you want to keep your data very like safety, keep it separated from other from the, let's say, the website. We have like local database downloaded from the next strain, and it's like completely integrated into the server. which means you do not need to share your data to the public. You do not need to share your data to the internet. All analysis can be done locally, which is very safety in case you have some like sensitive samples or some security reasons. And here is some like bigger, the bigger screenshot of the results. You can have a like more clear idea about the analysis. And this one is basic analysis, and this one is advanced analysis. We have a coverage across the whole genome, and it's quite convenient. You can target it. You can target into a specific part. Let's say if you want to see the detailed information from 4,000 to 6,000, you just like narrow down the bar to this part, and it will focus this part and give you more detailed information about the depths in this region. And also, I think for most of the COVID-related analysis, people more like to focus on the sequences and downstream analysis based on this sequence, like the genome sequence. The IGVD will give you some ideas about the clade information and the lineage information. And actually the clade information sort of links to the next strain. And like I said before, the next strain is totally integrated in your local server. So it will give you the tree information and some other informations like the lower one, the clade information. And you can have more ideas like how to use this website or how to use this system from the introduction of the website. And also there are some on the lower part, like bottom right, we will have a introduction about the names, like different type of names used in different websites. So you can have more idea about like why is 19A, another is B28. So this will give you a brief introduction. And also this is the like screenshot of the integrated local analysis system. You can visit this system by simply press the upper right part. It's like a bar plot and the line plot. You can press the arrow pointed button to get this picture, to enter this system, and you can have some brief ideas about the QCs, clade, and also the sequences along the whole genome. Like there are some black regions on the upper right, like the sequence reveal. There's some black regions, which is like which hasn't been detected in this sequencing. And other parts like the blue, it stands for some variants like SNPs and indels. Also the green, red, and other kind of variants. Different type of variants have different colors. And if you click the show tree, you will have the whole picture of COVID-19 cases like till now. And here is the tree, and like in this case, your sequence is like 28 or yeah, it's 28 and it's displayed here. You can have an idea and you can make like unrooted trees or other sort of trees by clicking the left buttons. And also the PANGO, this guy gave you an idea about another database, another system to give you a more detailed idea about like how many cases are uploaded to the database, and the introduction for each cases, for each clade, for each lineage. So you can have more ideas about this. But I just think this one is like website based. So you can visit this one on the website and to find your image. Like this one is B28, right? B28, you scroll down the page and you will find the B28 and some introduction like which country and how many samples submitted and estimated date, sort of these things. And the last one is the system upgrade. I want to highlight this part because if you want to upgrade your pipeline, you need to be connect it to the internet. Let's say to talk to the internet and find the packages from the internet. So if you want to upgrade your system, I suggest you connect to the internet. Otherwise, all things can be done locally, like I said before, which is very safe. Okay, that's pretty much for my introduction. Yeah, and do you have some questions about this?
Thank you, Dr. Wei Tian, for your presentation. And now we are in the question and answer sections. So as you know, and have already been typed in the chat that for you who actively asking questions, we have 3 rewards for each of you. So please ask as many questions as possible, as good as possible. So maybe you will be the awardee for this reward. So there are some questions that have already been answered by our MGI marketing team, and I would like to deliver a question that have not been answered yet. So one from Jack Ma to Dr. Yao. So the question is about the multiplex PCR-based sequencing. You mentioned it could be used for quantification viral load. So what does it mean? Could you please explain more about it? Opportunity, Dr. Yao. Hello? Hello, Dr. Yao?
Hi, can you hear me?
Yes, I can hear you.
I think I can help to explain this question. Because we have added some external DNA to our panel, so we can amplify both lambda DNA and the virus RNA together. And within the sequencing data, we can calculate the ratio of the SARS-CoV-2 and our lambda DNA to calculate a percentage of the virus, so that we Oh, sorry, I think I mute myself. So because in our panel we have we used some external DNA of lambda, so that we can amplify the all lambda DNA and virus RNA together. And within our sequencing data, we can calculate both the percentage of the lambda DNA and the SARS-CoV-2, and so that we can have a relative quantification of the virus. Hello.
Thank you for the answer, and I hope it answer your questions. So the next question goes to Dr. Ziswat. So if you do not include the Megabolt for downstream analysis, do you have any recommendation software for each application? For example, metagenome, SARS-CoV-2 genome sequencing, or WGS eukaryote cells. Yeah. Relative quantification of SARS-CoV-2 was achieved by spike in equimolar 200 copies of lambda DNA into each sample during library preparation, which co-amplified SARS-CoV-2. So do you have a answer for this, Dr. Ziswan? About the recommendation of the software for each.
The question is about. I answered the question for Zishuan that the question about the downstream analysis. Since our machine generated a standard format data FASTQ, which is universal data format, could be used for any open source pipeline, which compatible with other platforms such as Illumina. And also for SARS-CoV-2, we have open source pipeline has been uploaded to GitHub. And we also tested the link in the chat area. You can see for other software. Hi, can you hear me now, Sonny?
So you like to answer it in the chat box, right?
I add in the chat box, and also I mentioned that MGI data format is, we also have some partners for providing like the analysis, such as Congenica, UK-based analysis company, and also Sophia. They all provide the sequencing analysis service for such as WGS and cancer and other applications. And also we have the open source pipeline for SARS-CoV-2, and I will test the link here in the chat column.
Okay, thank you. Here I would like to translate you a question from our participant in Bahasa Indonesia. So the question is for the Mr. Weitian, yeah, the last speaker. So with the capacity of Etoplex, which reached to 3,000 samples per week, yeah, which the preparation should be conducted automatically. So she asked you about how much the cost per sample, how much the cost per sample that generate from it.
You mean the cost?
Cost. So the cost per sample. So which per sample? Yes, per sample. How much do we have to?
We will
spend the money for it.
We will separately reply it. Yeah.
Okay. So this from Kartika Devi. Mrs. Kartika Devi asked you about this cost of the per sample.
So we will separately send it to you. Yeah.
Okay. Thank you. So I think most of the questions have already been answered. Yeah. So I think there is a quite interesting question here. Since the mutation rate of SARS-CoV-2 is quite high, I would like to ask about the phylogenetic tree analysis for this system. Are there database system, updated database system for the new strain that found globally? So like, is there?
I think after you got the genome of the sequence of the COVID-19, you can use this sequence to do the analysis by yourself using the online systems or online databases. Alternatively, the next strain I mentioned before is which is integrated in our server, is like the latest version. So we always upgrade it. So you can sort of use the latest version of the database to fertilize your analysis locally or on the website. Both are doable.
Okay. Thank you.
All right.
Yeah. So there's also questions about specifications for the system. So do we need a specific place for installing the system? It means that should the room should be equipped with humidity or something like that, or can we only put the machine in a normal PSL2 or even PSL1 laboratory? Do you have information about it?
So that question is also for me.
Yes, Mr. Vietian.
Yeah, from my understanding, the megabolt. Actually, we have different kind of megabolts, like different type of servers. For some like very basic ones, I just think for the itoplex analysis, some basic one is already enough for the analysis. That one is quite flexity. So you don't need to make very strict environment for it. Just like
a refrigerated room, it probably enough exhaust or what is circulations. I think it's enough, yeah, or not. There's also questions. What is the goal of viral load measurement in COVID-19 diseases? So maybe it can be asked to Mr. Weitian. Can you answer the questions? What's the goal of viral load measurement in COVID-19 disease?
Hi, Dr. Tian. His network is not good. So as to the viral load measurement, we think the viral load measurement is kind of a part of identification, the purpose of identification. But the main purpose of our itoplex product is for the virus genome assembly. So it's for traceability and mutation analysis. But we can, for the quantification part, it's useful to identify the viral load, and for clinical use, like the activity of the virus, we can use for the clinical measurements.
Okay, thank you for your answer. So we are now waiting for the other questions. So I would like to inform you again that the webinar is recorded, and we will blast the information to the registered participant for this recording. And please feel free to contact us if you would like to ask additional questions to Mr. Personally to Mrs. Sylvia Sanchez, who's our product manager.
Yeah, as to the question previously about the system updated since as the new strains found globally the others, we will update the database if there's a new virus published, and we will update it to the database as needed. So it's not a fixed time. So since there's new strains being reported, discovered, we will update the database. If you want to know more about the MGI products and MGI SARS-CoV-2 sequencing solution, please visit us MGI website. I'll tap the website here at en.mgi#.com. You can check the more information about our products. We have the application notes and the data sheet on our website.
Well, I'm sorry. Can you hear me
Yes,
now?
you can.
Yeah, my computer just crashed.
Yeah.
I can't use it anymore. Yeah, like I said before, the very basic version of Megabot is can be used in the normal temperature room, and the humidity I think is also okay. But if you want to purchase some like more fancy ones, I recommend you use some like lower temperature rooms.
So do we need a PSL2 for this equipment? Something like a exhaust, and what is a low pressure room, something like that? I think it's not required.
I think it's quite different from the sequencers. For the sequencer, we need some like very strict environment, but for the server, I think just the very because the server is just like a computer. You can put it anywhere. You can put it next to your sequencer, or even in your office.
Okay. So how about the like calibration or something like maintenance? Do we have to do the customer have to perform the calibration every years or something like what is the annual probably regular maintenance? Can you inform us?
So sorry, can you repeat that again?
My question is about usually yeah, for example, from PCR or something, equipment like that, we require the calibration that conducted regularly and not properly to 2 years or 3 years. So for this MGI system, do we need to perform a calibration regularly, something like that? I hope you.
Yeah, I think it's more like related to the sequencers, right? This question.
So there
For
is
the server, I think it's you can upgrade it every time when you want to upgrade your system.
okay.
Hi, Sony. I asked Dr. Shen about if the sequencer need category. Before we install the machine, before shipping to the end user, we will do the category in-house, and our FAS will also FSE will do the check during the instruction, and after that, you use it. You should wash after you use. You don't need extra maintenance. And we have the standard maintenance procedure for the sequencer. FAS team will and FSE will share with the end users when they do the installation. And as to the laboratory environment, we recommend to use the BLS2 level laboratory for the nucleic acid extraction. But as to the library construction part, the normal PCR room is okay. And for sequencing part, is a standard laboratory is fine. So only the extraction, nucleic acid extraction part, we recommend you have the higher biosafety level.
Okay, thank you. And I have a question for Dr. Yao Jian. So our participant would like to ask you about which platform do the customer have to afford to buy to detect all of the COVID-19 variant available now and in the future. So probably he or she asked you about what is the best system that we have that she or he have to buy. Can you probably give a clue for this?
As to that kind of, because our sequencing platform, they all can provide the SARS-CoV-2 sequencing. The only difference between our sequencing platform like G50, G400, and T7, it depends on the sample amount. For example, if you precise like 4,000 samples, 5,000 samples per year, I think G50 is enough. And if you have more samples like 1,000, 2,000 samples per week, we recommended G400. And if you have a higher sample amount, even like 10,000 samples per month, and we recommended maybe T7. So it really depends on the sample amount. So generally, for the medium-sized lab, probably we recommend G50.
Because I think this question is quite interesting, yeah. So I would like to ask Mbak Sylvia. Mbak Sylvia, are you there?
Yes.
Mbak Sylvia, yes. Can you probably rephrase again the answer from the MGI marketing team, probably for those who not really have a understanding for this, for which platform that recommended for the COVID-19 variant available now and in the future? Maybe in Bahasa.
Yeah.
I have already replied
Oh,
it
yeah.
in the column. Can you check it? Sylvia, could you please check the answers in the column and help to translate it into the Indonesia language?
Oh ya, oke. Ya, jadi untuk MGI ini kita punya dua platform. Jadi ada yang G50 tadi yang sudah dijelaskan. Jadi G50 itu dia bisa outputnya maksimal adalah 150 giga basah. Kemudian yang platform kedua itu adalah G400. Jadi G400 itu bisa sampai 6 tera basah. Nah, kemudian untuk varian itu sebenarnya kita bisa menggunakan baik yang G50 maupun yang G400 tadi untuk deteksi varian. Jadi kalau di MGI ini dia dilengkapi sama yang namanya tadi Megabolt dan software yang tadi dijelaskan oleh Wejian. Jadi software ini adalah Autoplex software di mana nanti kita bisa menganalisis apakah dia mutasinya, kemudian bagaimana phylogenetic tree-nya yang dihasilkan dari hasil analisis sequencing kita. Nah, Jadi kalau untuk deteksi varian, terutama untuk mutasi, itu sebenarnya kita bisa memakai dua platform ini, jadi tergantung dari throughput-nya. Berapa banyak sampel yang mau kita kerjakan per minggunya atau per bulannya. Jadi kalau misalnya sampel kita high throughput, biasanya kita mengarahkan yang ke G400 atau G400. Kemudian kalau sampelnya tidak terlalu banyak, mungkin 96 per weeks, nah ini kita bisa mengarahkan ke yang G50 atau G50. Jadi keseluruhan dari presentasi ini adalah kami ingin menginformasikan terkait dengan workflow solution. Jadi satu workflow untuk COVID, mulai dari dia preparasi, kemudian sequencing atau NGS-nya, kemudian sampai kita mendapatkan report datanya. Di mana kalau untuk report data itu kita melengkapi dengan yang namanya software dan server Megabolt dan Atoplex software. Tadi seperti yang sudah diinfokan oleh pembicara terakhir, jadi dengan menggunakan software dari Atoplex ini, kita sudah secara otomatis akan mendapatkan detail data terkait dengan mutasi, kemudian terkait dengan clade, dan juga filogenetiknya itu seperti apa, font filogenetiknya. Jadi ini memudahkan, mungkin ada beberapa lab yang memiliki kendala di analisis NGS, nah ini bisa menggunakan platform dari MGI. Nah, tadi terkait dengan penawaran, mungkin nanti bisa menghubungi kami di SenseWorks. Jadi kami ini punya 3 skema penawaran. Jadi yang pertama itu bisa melakukan pembelian instrumen, kemudian kita juga melakukan penawaran untuk KSO, yaitu kerjasama operasional. Mungkin ada lab-lab yang ingin mengembangkan service, jadi aplikasinya itu banyak, nggak hanya untuk COVID aja. Jadi di next webinar kita akan melihat aplikasi-aplikasi yang memungkinkan direct report, jadi untuk analisis langsung. Jadi seperti yang saya sudah informasikan di awal, nanti kita next webinar itu akan ada pathogen fast identification, jadi bagaimana kita melihat atau mendeteksi semua mikroorganisme yang ada di dalam sampel. Jadi nggak hanya COVID. Jadi mungkin dia simptomnya itu seperti COVID, tapi ketika dia di qPCR dia negatif, kita bisa lihat dengan menggunakan PFI atau pathogen identification. Nah, itu kita bisa melihat patogen apa saja yang ada di dalam sampel tersebut. Nah, kemudian balik lagi ke yang tadi, ke skema penawaran. Jadi kita ada 3, yang kedua tadi kerjasama operasional, dan kita juga membuka peluang untuk key opinion leader. Jadi untuk para rekan-rekan peneliti yang memang aktif research, kami membuka kerjasama juga untuk sebagai key opinion leader, yang nanti mungkin untuk lengkapnya bisa ditanyakan di tim masing-masing. Jadi kami memiliki representatif area di tiap wilayahnya, jadi monggo silahkan ditanyakan ke tim sales kami, nanti kami ada proposalnya. Nah, jika berminat nanti bisa kita follow up, seperti itu. Ada lagi Mas Sony yang ingin ditambahkan?
Terima kasih Mbak Sylvia for your holistic answer. So we are now almost the end of our webinar, but there are some still questions. I would like to ask you one question from Mr. or Miss Oni Nurrahman. Talking about city value of the first question, I have different opinion. In my opinion, city value will affect the quality of the sequence that will be carried out by the machine. On the other hand, the number of the sample may affect the number of replicate and quality of the data analysis. Probably this question will be suitable for Dr. Wei Tian for the answer. Do you have comment for this, Dr. Wei Tian? So I would like to ask, deliver it again, the question. So talking about the city value of the first question, I have different opinion. In my opinion, city value will affect the quality of the sequence that will be carried out by the machine. On the other hand, the number of sample may affect the number of replicate and the quality of the data analysis. Probably Dr. Wei Tian can answer the question.
Hi. Sonny, I'll answer the questions. So because we add our quantity internal control in our kits, so if the virus loading level is very low, and comparatively our internal control is higher. So the reads count to the virus, it will be less. Therefore, we recommend if the virus load is low virus load sample, we need to generate more reads. Therefore, we can have more reads for the virus. Otherwise, the internal control rate will be higher.
Thank you. I think, and I hope it answers your questions. So we are at the end of this webinar. I would like to say thank you for your participations. And as I've been informed in the beginning that the recording will be blessed to you, to the registered participant, and also the presentation file will be delivered to you by email for the registered, also for the registered participant. So we would like to thank you for our speakers today for your great contributions. And I hope that this webinar can give you an outlook about the MGI system for next generation sequencing. Also, we are waiting for your feedback in the future. And in the behalf of the all committee, we would like to say thank you for your participation. Thank you. Ms. Sylvia, do you have additional information about it?
Not yet. Thank you.